Hi, everyone. I'm Ajay Shah, CEO and co-founder at Cytovale, and it's fantastic to have you here with us today for our first webinar of 2025, where we'll talk about the path for the IntelliSep Sepsis test from concept in the early days in an academic lab at UCLA, all the way through development, FDA clearance, and now commercial experience on market. At Cytovale, we're really focused on the early identification of sepsis from the first moments of a patient's journey, because we recognize that these first moments when people walk through the front doors of an emergency department saying, "I don't feel well today," are amongst their most vulnerable moments and amongst the moments where there's the most opportunity to make an incredibly meaningful impact in patients' lives and in healthcare outcomes for both patients and hospitals. IntelliSep is an FDA-cleared 8-minute blood-to-result sepsis test that focuses on looking at the quantification of the host immune response for patients presenting with signs and symptoms of infection to aid in the risk stratification of sepsis detection. Over the last 10 plus years, we've had the privilege of developing this technology, as I mentioned, from basic science all the way through commercialization, and wanted to take a few minutes with you today to share a bit about that journey, starting from the hard science of cellular mechanics, really all the way through the patient impact. Dino, do you want to kick us off and share a bit about yourself and the underlying science that drives Cytovale?
Thanks, Ajay. I'm Dino DiCarlo. I'm a board member and co-founder of Cytovale. My day job is a professor of bioengineering at UCLA and chair of the bioengineering department. Cytovale's IntelliSep test, the underlying technology behind that is what we call deformability cytometry, which is a technique that uses microfluidics to align cells so that one by one, so that we can then squeeze them to measure their deformability or how much they change shape. And we can do that at thousands of cells per second. Why do we want to measure how cells deform? Well, I think a good analogy is when you are looking for a ripe avocado. When you just look at it on the surface, you can't really tell if that avocado is ripe. But if you start to squeeze it, you get some insight into its ripeness, the structure inside the avocado. Very similarly for cells, if you just look at them, you don't see the information about their intracellular structure. And by squeezing them, we can get really quantitative information about that structural changes. The mechanics of cells, that's something that has been studied in the academic circles since the early 70s. Actually, one of the former directors of the National Science Foundation, Subir Suresh, had done a lot of work on linking mechanical properties of cells to disease and across many different diseases, from cancer to immune state changes. So this is a great marker of disease because it's a holistic change in the cell that is reflecting functional changes in the cell. The problem over in this kind of original work was that the ways that we measured cells was very slow. So you can maybe measure one cell per day using a pipette. And now with deformability cytometry, we're able to measure tens of thousands of cells per minute. And what's really exciting is that these measures of cells, these mechanical changes that reflect structural changes, these are things that can directly link to immune state, immune activation, and sepsis, unlike things like the genetics, transcriptomics of the cell, which are kind of later indicators or may not indicate these changes at all. So, as we started to use deformability cytometry in the lab, and this is started in the lab where Henry was a student and working on this technology, we kind of applied it to a number of different areas: regenerative medicine, cancer, basic biology, as we looked at nuclear structural changes. What Henry really found, which was really exciting, though, is that we could see signatures of immune cell activation, and this formed the basis of development of the IntelliSep test. So I want to pass it over to Henry to share more about really how you advance the science from UCLA in the early days to that kind of bringing that further in the company.
Thanks, Dino. Hello, my name's ernurse. I'm the CTO and co-founder for Cytovale. It's been quite a journey since 2013, bringing the core microfluidics technology out of UCLA and MIT into the clinical diagnostics for sepsis. Right? The IntelliSep test is a convergence of the microfluidics that Dino talked about, the ultra high speed imaging that we utilize, and AI that allows us to interpret this really large data set that we capture. I think importantly, boiling it all down to a clinically actionable score for our customers. To start off, right, we talked about the IntelliSep utilizes microfluidics. These channels are microchannels, the width and the size of your hair. Gino mentioned we sample at over 1,000 cells per second, really giving us that high statistical confidence to be able to look at heterogeneity biology, to be able to infer complex disease states. And what's really impressive about the device is cells are traveling at many meters per second. And they change, they're forced to stop and change direction, much like in a four-way intersection. Forced to stop and change direction, and they're subjected to thousands of G-forces over a very short amount of time, about a millionth of a microsecond. That's where high-speed imaging comes in. The high-speed imaging that we utilize samples at over 500,000 frames per second and totaling to 8 million frames for every sample that we look at. And what this information is capturing is shape, size, dynamics of the cells as they're transiting through the junction. And what this allows us to do is capture the mechanics and observe the mechanics of the cells as they're being subjected to these fluidic forces. So I think what we're measuring with respect to sepsis is leukocyte activation. And with leukocyte activation, I think importantly, the epigenetic state of these cells. One mechanism that's well known in sepsis is apoptosis. And this is where the neutrophils go through a specific biological cascade where they take their tightly wound up DNA and they expel it outside their cell body in a response to the cytokine that they're receiving. This response has been shown to be linked to organ failure and ultimately down the road of sepsis, sepsishock, and mortality. So for this event of going from tightly wounded to fully decondensed and now outside their cell body, is a massive structural change of the cell. And that's the key insight and hypothesis that Cytovale is measuring. IntelliSep test is measuring the activation of leukocytes, and specifically these activations of these innate immune cells. The last part of our convergence of technology is the AI that we use to make sense of the information that we're collecting. We talked about a really rich data set that has many million frames. It's both temporally rich, spatially rich. So we use deep nets to help us extract features of interest that quantify the mechanics of the cells. We then apply machine learning to this hyperdimensional space to help us refine the feature set to ultimately boil it down to an IntelliSep score. So sepsis is a really complex disease. I think there are many pathways that lead to sepsis, and there's many kind of the pathobiology of sepsis. There's many ways to get to that mortality and morbidity. So having a direct measurement of the biology is a key to, I think, better understanding the disease state.
Yeah, and Henry, if I could jump in there, right? I think what's really cool is that over the last decade that you've been developing and then kind of expanding on the basic science and the technology work, we've seen the sepsis field evolve, and really within that, even the definitions of sepsis, right? And I think what's been really cool is to come to this moment where we have a convergence of the broader kind of medical understanding that, sepsis is not simply an infection, right? Sepsis really is, the body's, dysregulated immune response to an infection. And what we do with IntelliSep is directly see that dysregulated immune response, starting at a single cell level and as you said, putting together that picture across, tens of thousands of cells. But I think what's equally important is the technology work that you did to develop this into a test that can be clinically actionable, right? And that meets that patient where they need it most in just the first minutes of their patient journey. Can you share a bit about some of the practical workflow elements?
Yeah, sure. So IntelliSep is a rapid test. It has an 8-minute turnaround. So from blood to answer, it's 8 minutes. Additionally, the overall hands-on time is 9 seconds from when the initial operator interacts with the instrument to when the instrument is then automated to complete the test. And lastly, the test uses 100 microliters of blood. This is from a purple top vacuum chainer. It could be the same draw as a CDC and really simplifies the mutation process. And we talked about the end result being the IntelliSep score. That's also broken down into interpretation bands. Low likelihood of sepsis and a high likelihood of sepsis and in between. And this gives indication of severity of sepsis as the bands increase. So this direct measurement of sepsis is resulted really at the time when the CBCs also returned, right? And this gives the providers more information to have that early differential diagnosis. So I'm glad that the applicability of sepsis is not just theoretical, and we've seen a lot of success with our customers and the value realized when implementing this test. I think Jay can share more there.
Yeah, and so over about the last, little over a year that we've been commercial with IntelliSep, we've had the privilege of making an impact on over 30,000 patients to date, with over 30,000 tests commercially run to date. And with that, are able to look kind of beyond the basic science and validation of IntelliSep to really starting to understand what are the clinical outcomes that can be driven by enabling early sepsis detection at the beginning of the patient journey when they first present to an emergency department triage desk. And what we've seen is pretty incredible. Staying first within the emergency department itself, we've seen two compelling clinical scenarios play out. One is when there are patients who just don't look that sick and look stable and are often sent back to what is a busy ED waiting room where they could sit for hours. And then seeing that when that IntelliSep test comes back in band 3, it allows the emergency department to recognize that even though this patient might not look that sick, they really have sepsis brewing in their veins, and allows them to go and pluck them out of the waiting room and accelerate their care by well over an hour, driving to improved long-term outcomes for that patient. And then at the other end, when there are patients who are very clearly sick, very clearly in shock, oftentimes the emergency department, rightly so, focuses on sepsis and looks to accelerate sepsis treatment. And when IntelliSep in those cases comes back with that band 1 low-risk result, it enables them to see that, hey, even though this patient is clearly in shock, there might be something else driving their basic biology. And allows them to pause and pivot, and they're often finding cardiogenic causes of that otherwise undifferentiated shock presentation. And so those are, examples of just the patient clinical wins we're seeing in the emergency department. From an operational perspective, hospitals and health systems that have leveraged IntelliSep have shown a meaningful ability to, improve their time to disposition for these patients, their ability to improve their nursing and provider confidence in delivering the right treatments to the patients, as well as kind of global quality metrics like a double-digit improvement in their SEP1 scores. Going beyond the emergency department, what we've seen is that by using IntelliSep upfront to provide accurate risk stratification for likelihood of sepsis, you really enable the patient to get on the right pathway and reduce clinical anchoring. And in doing that, are able to actually reduce length of stay by about a day for these patients. And all of that translates into about a $1,400 cost savings per patient tested. And then finally, I think the thing that we're most proud of at Cytovale is the lives we've been able to save. And so in early data coming out of partnership with our launch partner, they were actually able to demonstrate an over 30% reduction in the risk-adjusted mortality for their sepsis patients. And so for their, busy regional emergency department, that translated to about a life saved a week. And that's one more patient going home to their family. And that's something that I'm personally really proud of. And I think we as a company, take a lot of pride in being able to make a meaningful impact and help our hospital partners help their patients. And so as we take a step back and reflect here, Dina, would love for you to bring it full circle and share some thoughts on kind of this arc from basic science to patient impact.
Yeah, I mean, as you hear what Ajay is saying in terms of this huge impacts on patients, hospitals, doctors, like this is something that makes me extremely proud to see, technology that started in our lab have this huge impact. But I should definitely make sure that I give credit to the federal funding that helped start this work in the lab from the National Science Foundation, Defense Advanced Research Projects Agency. This is DARPA. I know that had also been involved in their work in initiating the internet, right? As well as the NIH. So yeah, great. From basic research all the way to clinical impact, amazing for us. But I'm also extremely excited by how now we have this new tool that really links to the fundamental biology of sepsis. And this is the first test of its type that is measuring these mechanical properties of cells and has the opportunity to help us define sepsis in terms of this innate immune activation that we can now measure and quantify for the first time. So this is what makes me extremely excited because, as we've gone through these clinical studies, we realize that this approach generalizes across a number of different sources that are causing this immunoactivation, whether it be bacterial, viral, et cetera. And so that conserved biology is something that we're measuring for the first time. And it opens up so many opportunities. So really excited to see where we can go from here.
So, I know we spent the last, bit of time here talking about the science and technology behind IntelliSep. And obviously, we're proud to be innovators at the forefront of this journey. Want to share with the audience that at Cytovale, we recognize that sepsis is a complex and emerging space, and there's implications here for patients, for providers, for health systems that span all the way from the basic science we've been talking about through the patient journey and all the way through some pretty meaningful things like even revenue cycle implications. And so we recognize that technology is just part of the story, right? To drive real meaningful change in healthcare, it takes a combination of not just science and technology, but also people and process. And to that end, as we've been advancing IntelliSep into the clinic, we've actually had the chance to build out our team pretty meaningfully to include, former laboratorians, former ED nurses and the like. And some of the most exciting conversations we've been in recently have been in partnership with health systems, where we can together understand what are their specific needs and their specific use cases, right? Which are the health systems who are best poised to leverage IntelliSep and embed it into their upfront triage workflows to help answer the question, when the patient comes in at 2 a.m. with a cough, is this a medical emergency? Right? Do they need the accelerated care for sepsis? Or is this a patient that can be safely managed in alternate settings? And so helping provide an insight to aid in the diagnosis of sepsis and guide health systems through that journey is something that we're really passionate about. And so we'd encourage those who are interested in, picking our brains about the sepsis space, about the technologies out there, and in exploring how we can potentially partner together to reach out. But with that, really appreciate the time to share the story with you all today and happy to take your questions.